Continuing with the Life Learn series of our Science Reporter magazines of November 2022, the next topic is magnetotactic bacteria. The magnetotactic bacteria gets their name from magnetotaxis, or the ability to align and coordinate along the Earth's magnetic lines. They build nano-sized magnetic crystals called magnetosomes in their cells, which act like tiny magnetic compasses. They use the resulting magnetic moment which is generated from these magnets to steer them towards favorable living conditions in their habitats. The scientists are still deciphering the cellular mechanisms of how these bacteria synthesize these nanocrystals. So let us delve into the world of these fascinating magnet manufacturers and get acquainted with them. Let us explore their world. Magnetobacteria or magnetotactic bacteria are prokaryotes, single-celled microorganisms without a distinct nucleus or well-defined organelles in their cells. Instead, their genetic material floats about the cytoplasm. These bacteria live in freshwater and marine habitats in oxic and anoxic regions, that is, where there is an oxygen gradient in the water. As anaerobes, they do not require oxygen for cell functions, and they thrive in oxygen depleted zones in the water sediments. The bacterial cell tapers into a tail-like extension called flagellum, which helps the organism swim around in its habitat. The magnetotactic bacteria are expert swimmers, clocking double the speeds of E. coli bacteria. Blakemore found that each bacterium contains several nano-sized magnetic crystalline structures called magnetosomes. The magnetosomes are aligned in a chain-like manner along the central axis of the cell. When shrunk together, the chain behaves like a magnet with poles. This living compass orients along the Earth's magnetic lines. The research has shown that due to the magnetic moment, a magnetic torque develops in the cell, which gives a push to the flagellum to rotate it clockwise or anti-clockwise. This rotation propels the cell's movement. Magnetotactic bacteria are often found swimming from high to low oxygen gradients in their environments, northwards in the northern hemisphere, southwards in the southern parts, and in both directions in the equatorial regions. The cutting-edge technology. These bacteria are skilled engineers employing complex molecular gymnastics to synthesize magnetic nanocrystals. Iron is the chief element for making them. Iron plays a vital role in several metabolic pathways in organisms and magnetotactic bacteria. They too require this essential nutrient to grow and proliferate. However, iron is not available in a stable state in their living environments due to the lack of oxygen. In aerobic or oxygen-rich environments, iron is present in the ferrous state, that is Fe2+, which quickly gets oxidized to the ferric, that is Fe3+ state. Therefore, these bacteria have devised an alternative iron absorbing technique. Research has shown that these bacteria release particular ion binding molecules called sideropores into the vicinity. The sideropores have a high affinity for Fe3 plus ions and latch to them, after which they are absorbed into the bacterial cell. Once inside, the ferric ions are cleaved from these molecules. Studies indicate that synthesizing magnetosomes from the absorbed ion involves several steps, some of which remain unclear. However, broadly, the bacteria undergoes the following steps. They absorb the ion, they form a biovesicle in the cytoplasm, they transport ion into these vesicles and biomineralize the ion into magnetic nanocrystals and then into magnetosomes. The master maneuvers. Over the decades, scientists studied these bacteria in a greater detail to understand how these living magnets function. They found that the bacteria uses the magnetosome chain to decipher the orientation of the Earth's magnetic field. The chain generates a magnetic field of about 0.5 Gauss, creating a magnetic moment or energy in the organism. As the magnetosome chain is stacked along the central axis of the cell, a torque develops which they use to push the flagellum and rotate it clockwise or anti-clockwise depending on the orientation. Thus, the flagellum is steered and this propels the organism to move. Depending on which hemisphere the bacteria live in, they swim up or down in search of anoxic regions. Research has found that bacteria exhibits photokinesis or increased swimming speed in red light ranges and slowing down in green light conditions. However, they avoid intense UV light regions. Myriad shapes. The researchers observed magnetosomes under an electron microscope and found them to have 2D or two-dimensional uniform crystalline structures. The MAM gene, which is responsible for forming the membrane proteins and cytoskeleton, the magnetosome's membrane proteins determine the morphology of the crystals. 4 types of MMS proteins are involved in the crystallization process to form these nanostructures. Different species make different shaped crystals: rods, spheres, cubes, elongated octahedrons, and cubectohedra and prismatic. Researchers have isolated the magnetosomes from the bacteria and found them to be highly crystalline and stable particles of 30 to 100 nanometers. Unfortunately, our most advanced technology cannot still reproduce these crystals' structural finesse and stability. However, science has found that magnetosomes have potential in targeted therapies, especially for cancer. Hence, they are mass producing magnetosomes growing bacteria cultures. The key players of Earth's ion cycle. Magnetosomes are stable particles and they retain their structure and composition even outside the bacterial cell. The magnetotactic bacteria consists of up to 3% ion by dry weight. Studies have found that magnetotactic bacteria plays a very crucial role in ion recycling, replenishing up to 10% of the mineral in the environment. When the bacteria dies, magnetosomes deposit in the sediments as magnetofossils or are ingested by other organisms, completing the cycle. Nanobiotes ahoy! Several experiments show magnetosomes have the potential in new age cancer therapies as external magnetic fields can manipulate the magnetosomes. They are non toxic to the humans and do not degrade in the body fluids. Researchers propose that magnetosomes can be injected into the body close to the tumor region, guided to the cancer site with external magnets, and at the site made to perform cancer inhibiting actions like localized radiation or hypothermia. It applies a safe level of alternating magnetic field. The magnetosomes will rapidly align and realign to the field. The nanoparticles expend energy in this process. Dissipated heat cells kills the cancer cells. Localized chemotherapy manipulates the outer layer of magnetosomes coat with drugs, steer to cancer site, unload drugs directly to the cancer cells.